Okay, hi everybody. It is 1:15, so we should get started. Can everybody hear me okay? Okay. So there's been a few comments in the chat section about the lab report. So I was thinking maybe that would be a good place maybe to start, see if you have any questions. I mean, I can't like look at each of your lab reports individually right now, but certainly we can talk about questions you've got. So I'll open the floor, and Victoria, you want to ask any questions or anybody? You can all hear me, right? You're just all being
Is
shocked.
there a certain, oh, sorry.
Go ahead.
Is there a certain amount of citations you'll want us to have? I only have one citation. I formatted in the APA format. I wasn't sure how many citations we need.
Well, you know, I think
if we need a certain amount.
I think 10's plenty. I'm kidding. No, you don't need 10. Well,
I was gonna say I do not have that.
Well, you have the lab book, right?
Yeah.
That's the one you're talking about. And everybody should certainly have that. But if you go online, let's say, and look up information that you want to put into, let's say, the introduction section, where you talk about the scientific method, the lab book has a little bit in it, and we talked about that. But if you wanted to expand upon that, or if you wanted to expand upon the practical value of this sort of a study, right? We talked a bit about that as well.
Why people just don't talk.
Yeah.
Type in the chat. Okay.
You would want to cite.
Turn on the microphone for 2 seconds to talk. I pretty much know what's going to happen today.
Hey, Caitlin.
We had review. Oh, hang on.
Caitlin.
Oh.
Can you mute? Thank you.
Oh, I'm so sorry.
Right, no problem. You would want to make sure that if you used outside references, you know, be they online or what have you, that you cite.
Yeah, that's what I did.
So I would think that perhaps some of you might want to explore, you know, beefing up, if you will, say the introduction or what have you. And you know, I'm not saying you need to do exhaustive research, but I was talking to Kylie earlier, and it's not that I'm expecting you to do tons and tons of online research and have your introduction 5 pages long. That's not what I'm saying. But pretend you're the instructor and you're reading somebody's lab report and you see that someone's really put an effort in trying to make this introduction, you know, a good one or whatever. That's going to impress you, right? It's going to indicate that the person's gone beyond the minimum. So just give it some thought. This isn't rocket science. It's like, you know, show a little initiative, and that's not a bad thing.
I only had one citation that I really like. I found a website source. I cited it in the APA format. I just wasn't sure. I don't know. I'll have to go back through. I might add more details so I can cite. Because I only have like one citation in my works cited page, so I wasn't sure if I should add more. Maybe. I mean, I think my lab is full of detail, but I didn't really, I don't know. I'll have to go back through it.
The goal
we'll just see how you.
The goal is not to get a ton of citations or references in an effort to impress the teacher, right? I'm not saying that. But,
I know it's instructional, more less.
Yeah.
Kind of explains what the experiment was.
Any practical value. What is the scientific method? These are things you can put in the introduction. And I think there's online stuff you can find out there on that sort of stuff. How about other questions related to the lab report?
I just had a quick question. Do we have to write all of the different steps on different sheets of paper? Like, because that's what it says in the instructions. I didn't know if you had to do that.
Where did it say that?
It just says, like in each of the sections, it says to write it in the heading of the left margin on the next page.
Right, that's
Like
the
through every section, it says that.
well, each section, by section I mean introduction is a section, materials and methods is a section.
Yeah.
Yeah, so those do not have to be on their own pages, no.
Okay.
You can have the introduction and the materials and methods all on one page, theoretically, depending upon how big they are.
Yeah.
But
Okay.
I said the title page and the list of works cited, those should be on their own pages.
Yeah.
Yeah. Other questions on the lab report?
I do have one question for the graph.
Okay.
As it shows in the results, it just says like table 1 or graph 1. Would that be an okay title for the graph? Just graph 1, or does it need to be like, should I add more detail in the title than just graph 1?
You know, you could call it graph 1, figure 1.
Yeah, that's what I called it, figure 1.
Yeah, and something like that would be fine. And
Okay.
since you only have the one table and the one graph, I would just refer to them as, you know, table 1 or graph 1. Yeah.
Okay, when we print them out, are we just gonna staple them all together then? Or should I make another copy of this graph? Or can I just staple this with my lab report? I mean, I'll end up getting it back anyways, I'd assume, unless you keep them.
No, I'll be giving them back to you. So include the results table and the graph right in the
Okay.
lab report. Yep.
All right.
You can staple them all together. I think there's a stapler in the back of the room too, by the way. I think there's one in there.
Okay, I'll do that tomorrow then.
Mm-hmm. Great. Any other questions on the lab report?
Sir, this is Ryan. Can you hear me?
Yep.
Sir, I just wanted to check on the results section. So you said basically just that we're just getting to give a short description of what the results were, and like I guess you would say like see table 1, see figure 1 in the actual like description. So your results section really isn't going to be long if all we're doing is it for a general discussion, and then all the discussion section is going to be done down in the actual discussion of the portion of the report.
Right. So Ryan, really for the results, there's not a lot of verbiage or writing you need to do for that, because the data table, the results table is what it is. It is the results of the experiment we did. So you do not feel the need that you need to write a lot of stuff there for the results. You can just present it in the form of the table and say something like see table 1 or see results table for data collected or something to that effect. You're right. It's short and sweet. Nothing.
Just a short blurb. That's all you want then.
Exactly. Yeah, exactly. The discussion is going to require some writing and some analysis and some thinking and such. Yeah. Because in that section, you're going to be talking about, do the data support the hypothesis? Yes or no, and why? And offer some suggestions as to why not, if indeed you're going to say that.
Right. Thanks.
Yeah, absolutely. Anything else on the lab report? Okay, I sent you guys an email today just reminding you to make sure you bring the appropriate handouts for lab, right? And for tomorrow, you know, there's a couple. I think there's maybe the one on the rat dissection that you should definitely print off and bring with you. I don't think there's anything on exercise 3 in Blackboard. I don't believe so. And some of you don't have your lab books yet. Is that true? And you're going to need exercise 3 from me?
Yes.
Okay. So how many people? I know, Ashley, you told me that the other day. I'll have some exercise 3 handouts on the microscope for you.
Okay, thank you.
Okay.
If need be, my friend who went to college at Pitt gave me their hardcover human anatomy book. So I have this loose leaf someone could also use too if needed. I can bring both books if that's, I think that's what we're talking about, right? The text.
Well, I was talking about the lab manual. Tomorrow's lab. You're talking about the
Oh yeah, the textbook. Yeah. Yeah. Well, I'm just saying in reference to, I have that if anyone needs it.
Well, thank you, Steph. That's nice. I let somebody borrow an older edition that I had of the textbook too, because her books haven't come in yet. Don't forget that the library does have copies of the textbook, but you can't check them out. So the sooner you can get the lab book and the textbook, the better. When I say text, I mean the lecture text. Yeah. So again, just to reiterate and remind you, tomorrow in lab, we've got the lab quiz that we're going to take first. Then we're going to do the lab exercises, and then we're going to finish up with the lecture quiz over chapters 1 and 3 in the textbook. Right? Everybody knows that.
And we were supposed to read up to page 108 for the textbook, correct?
I think we were going to put all of chapter 3 on the quiz. Did I write you guys an email indicating that?
Yes.
Right. So all of chapter 3, which by the way, all of the lecture, the Zoom lectures are posted in Blackboard for chapter 3. I will say that there was a slide or two at the very end of the PowerPoint that I failed to pull up when I did the Zoom. But if you look at the PowerPoint, and I can do that right now to show you kind of what I'm talking about. If I share my screen and I go to the chapter 3 PowerPoint, do you guys see the cell, composite cell diagram?
Yes.
Okay, so I think what I did was, if you watch the Zoom presentation, I ended with this slide. And I forgot to include these last 2 slides. This one has simply got a link, a Khan Academy link, which is really good. It covers these various topics that I talked about in chapter 3. And then I ask you at the very end here to check out the section there that describes what's called apoptosis. There's a short little section. It's like a 3-paragraph section there at the very end of the chapter that I don't think I spent much time talking about during the Zoom lecture, but I just wanted to draw this to your attention. These last 2 slides, this one and this one, did not appear on the Zoom recording. I ended here, and that was my fault. I just forgot. I just created these actually that day, and I forgot I had put them at the very end. So maybe what we could do is open up the floor for questions related to chapters 1 or 2 or 3. 2 is the chemistry chapter, right? of which we have the chemistry 30-point multiple choice quiz next Thursday. And 1 and 3 we've put in Zoom, the Zoom recordings. And hopefully by now you've had a chance to get into chapter 3. So again, I'll just open it up because we decided last week that we were going to use this time not to so much lecture as to open it up for questions and review, and we can go over the PowerPoints or whatever you're having any problems with. Well, I guess you're all fine with chapters 1 and 3 then.
I have one question. I mean, I'll have to look. I made a bunch of flashcards because that's how I remember things the easiest, I guess. Is the fibrous pericardium the same as the parietal pericardium? Or are they different?
They're different.
Are they similar?
Yeah, I'll tell
Would
you
you say?
I'll tell you the difference. Okay.
Yeah, on this one flashcard I wrote parietal pericardium and the outer layer, the pericardium. And for some reason I wrote same as fibrous. I'm not sure if I just didn't understand. I'm not sure what I did there. So
Okay.
I guess I'll just ask about that.
Yep. I'm looking for a good diagram to show you. And this is in actually chapter 15 on the cardiovascular system. And where did I see this? Okay, here we go. All right.
What page are you on?
I am on page 562, but I can pull this up and show everybody. So let me do that. All right, I have got to. I'm going to go to cardiovascular system PowerPoint. I'm going to stop share screen. And
Yeah, I mainly did my flashcards mainly out of the textbook. I'll have to take a look at the PowerPoint. So
are you guys seeing the cardiovascular system PowerPoint?
No, we're looking at Blackboard at the moment.
Okay. How about now?
Yep, the PowerPoint's up.
Okay. So this is a chapter we're not going to get into for a while, but there's a diagram right here. Do you see the heart wall or no?
Yep.
Okay. So this is a cutaway view, obviously, of one of the ventricles, and we're expanding in here. So in the lab book, where it talked about the various membranes surrounding the heart and lungs and so forth, it's very similar to this. So here's the heart muscle in red. This is the ventricle wall. And just on the very surface of that is what is referred to as the visceral pericardium. You might remember labeling that in, I think it was exercise 2 of the lab book. Okay? Everybody remember that? that visceral pericardium, it ends up folding back upon itself and then forms what's called the parietal pericardium, which I'm outlining here. This very thin, kind of grayish line, obviously labeled. In between the parietal pericardium and the visceral pericardium, which touches the muscle, is the cavity, in this case called pericardial because it's the heart. And as it says here, it contains what's called serous fluid. And we haven't talked much about this, but the book I think does describe it, or will soon. This is a lubricating fluid because the heart is beating in this sac. So getting back to your question about what is the fibrous pericardium, this fibrous pericardium is shown here in white. Okay. In other words, it's exterior to the parietal. So when we talk about like the sac that surrounds the heart, and you're going to see this when we do the dissection in another 3 months, most of that sac is what's referred to as fibrous pericardium, but the very innermost layer of it is parietal pericardium. Now, I don't know that the lab book even mentioned fibrous pericardium. I don't think it did. It had you distinguishing parietal from visceral. But as long as you brought up what is the fibrous pericardium, it's external to the parietal pericardium. You can see they're connected together. Okay, and then there's this theoretical space that contains the serous fluid, and then you have on the surface of the heart itself, the heart muscle, the visceral pericardium. So visceral membranes, we said, are actually touching the organ, while the parietal membranes are lining the cavity within which the organ is found. So I don't know, did that help, Steph, a little bit?
Yeah, it did. Now that I'm looking at the diagram, I've just been busy making flashcards on the other notes, but that was just a question that kind of stuck with me.
Yeah,
I got through the flashcards. Thank you.
That's going into a little bit more detail than the lab book, you know, wanted you to get into, but as long as you ask
That's
about
fine.
fibrous, yeah. And certainly we can talk about lab stuff here as well. I don't want to exclude lab, even though I mentioned chapters 1, 2, and 3. So it's an open discussion. Anything you want to ask about lab or lecture, now's the time.
I just had a question, like kind of general question. Do you have any like tips or tricks to help us remember the cell cycle, like all the little ones like within the mitosis and such like that?
No, not really. There's no magic bullet or pill to take. I would have thought that a lot of that you have already heard of, so it shouldn't have been totally new to you. They do talk about that in high school bio. No, I think you just have to look at the different, stages, for example, of mitosis and know what is taking place. There's some good videos. I think the Khan Academy is worth watching. That's excellent. Flashcards are certainly worthwhile for that particular topic because you can make a flashcard on interphase and one on prophase and one on metaphase, and include maybe your own sketch as well as a bulleted listing of the major processes taking place during those different stages. I think that
Yeah.
works out very
It's
well
like-
in note cards.
There's just such like the little ones inside, so I wasn't sure if there's like the inside the mitosis. There's like the 4 different stages. Like I know what each does. It was just like remembering them in like the order of the 4 little stages inside of it was all.
Well, I mean, you could always take the first letter of each stage, so P, M, A, T, and come up with a saying, right, to help you remember those. A little mnemonic. I don't have any off the top of my head, but you could make something up.
Okay, thank you.
I mean, those are the sorts of things, those are the sorts of learning techniques, right, that have been used for a long time. And what I'm thinking primarily about is like the cranial nerves, which you'll be learning about next semester. There's a particular saying that helps you remember those particular cranial nerves. But I have never heard of one. Go ahead, Ailey.
I kind of have something to help remember, like PMAT. That's my anagram, PMAT.
Yeah.
Anaphase, like it starts with an A, and the chromosomes move away from each other. M, they line up in the middle. Metaphase, middle,
Yeah.
and then telophase, they form like a telephone kind of, because there's two new membranes. So that's how I remember it. If
Good
it
for
helps.
you. Yeah, I like that. Good for you. That's great.
Thank you, Haley.
Yeah. Yeah, that's really good. I like that suggestion. So yeah, we should be sharing that.
On the quiz, I don't know if I'm allowed to ask this, but is it pretty much going to have like the diagrams and you pretty much just label it, or can you really give out much information?
Sure.
Like, is there going to be a lot of diagrams we're going to have to?
There's going to be a couple of diagrams. There's going to be a few. There's not going to be, however many there are in exercise 2. I don't know how many there are there, but there's not going to be, how many diagrams we got? 1, 2, 3, 4, 5, 6, 7. There's like 7 or 8. You're not going to have 7 or 8. You might have 2 or 3. And remember, the quiz is not strictly on just exercise 2. You could see a question on the lab experiment we did too. But because you've just written that lab report, that is like fresh in your mind, I would hope. So it's not like you really have to do a heck of a lot of studying for exercise 1. You did that. You wrote the report. That's going to be a great, asset in terms of that material. So you'd be good in terms of spending your time focusing on 2 more than 1, because there's so many more terms there. So to answer your question, Steph, yes, there's going to be a couple of diagrams. I'm going to pick out. So for example, here's the anterior region with variety of terms. I'm not going to name all 28 terms on figure 2.7. I'm not going to do that. But I might say, tell me what structure number 7 and number 22 are. And I'm going to give you a word bank for that particular diagram, because there's so many terms. Now, I'm not giving you a word bank for every single question on the quiz, but for those that required, that's a lot of terms to remember, and I understand that. So I'm going to give you a little assistance by giving you a couple, choices there.
This quiz tomorrow, is it on chapters 1 and 3, or is it on 1, 2, and 3?
Which quiz are we talking about?
The one tomorrow, the 10.1.
There's 2 tomorrow.
Okay.
Right? You all know there's 2 quizzes tomorrow. This should not come as a surprise. We have the lab quiz, which is over exercises 1 and 2. And then you should look over what we're going to be doing tomorrow, which is what? What are we doing in lab tomorrow?
Rat dissection.
Rat dissection and what else?
Microscope.
Care and use of the microscope.
Right, the microscope. So that's one quiz. The other quiz is the first lecture quiz, and that's over chapters 1 and 3 in the textbook. There will not be any questions on chapter 2. That is a week from Thursday. Everybody clear on that? Okay.
I just have something I'm curious about. For in the cell, when the checkpoints sees, and that's like for cancer, is there any other alert that your body gets besides like increased white blood cells, a lump, or feeling sick? Like, is there any alert that goes to your body that this has happened?
I'm not sure how to answer that. The answer is yes. So for example, if you were to have an injury, let's say you broke your leg, broke the bone, damaged some blood vessels. Now we know that when that happens, eventually we have to generate more bone cells, and we have to generate the process of repair in that fracture, right? But think about what other symptoms you would experience having broken your leg. Some of you have broken bones. What did you feel? Did you feel good? No, you felt terrible. It hurt. Did you run a fever? No, maybe not. In other words, the body does send out signals, if you will, to indicate that there's been tissue damage done. And that also, is telling you that the process of repair is going to be starting, not at the moment of fracture, but maybe, as time goes on, that process of repair is going to occur. The cell cycle is going to kick in, right, to produce more bone cells and whatever other tissues may have been damaged. So I think to answer your question, yes, there are alarm signals that get sent out in response to tissue damage, for example. If you get a paper cut, we know that in a few days that repairs itself, and you didn't even know where you made the cut, right? That's how quickly and that takes place. So in that sense, I don't know that there's a lot of alarm signals sent throughout the body because it's a less severe injury. But understand that whether you get a green light or a red light, and we're talking about control of the cell cycle, those green lights and red lights are dependent upon lots and lots of different chemicals and the expression of different genes within the cell. It gets complicated. I'm not sure that I answered your question, but I guess it depends upon what the example might be. So how about chapter 1, the introduction to A&P? Are we good with that chapter? Any questions about that? Any questions about homeostasis? The role that receptors play, control center effectors? Any question about that? How about chapter 2, chemistry chapter? Usually there's tons of questions on chemistry. Don't be afraid to ask. You really should be in chapter 2. If you haven't started that, you better get in there soon. Because it covers quite a bit of information.
For like the elements and stuff, are there going to be, are we going to have to like figure out like the atomic number, or protons and neutrons? Like, do you have any questions affiliated with that on the quiz? I wrote down like some tables from the textbook.
Well, I think you need to understand that if I gave you the atomic number of an element, let's just say oxygen.
Yeah.
If I told you the atomic number, which is 8, for an oxygen atom, could you tell me
That
how many protons are in the nucleus of that atom?
it would be the same, I believe, right?
Right.
As a atomic number.
Yeah.
Well, I'm just asking, like, in general. I wasn't sure how to ask that question.
Well,
But
that's what I'm saying. I could give you the atomic number of an element and ask you to tell me
Yeah.
how many protons there are. I could ask you how many electrons are there, which would be the same number. And could you tell me the number of neutrons? If I told you just the atomic number, the answer is no. What would I need to know before I could tell you the number of neutrons? I need to know two things. I need to know the atomic number and the atomic weight of the element. And how do you calculate neutrons? You take the atomic number and subtract that from the atomic weight.
Yeah.
So in the case of oxygen, the atomic number is 8. The atomic weight is 16. How many neutrons?
8
Right. Same
for
number
oxygen, right?
Same number as protons and electrons. But it's not always the same number, right? It depends.
Right. Yeah. It depends. All right. I was just curious.
Yeah, no, that's a great question. I'm not expecting you to know the periodic chart or anything like that, but I think you should know what is the atomic number represent, what is the atomic weight or mass number. Some books will use that term instead of atomic weight. Those are important things to know because they tell you a lot about how many subatomic particles there are in the atom of that particular element. And that's important because it tells you a bit about its reactivity in terms of forming bonds with other elements, especially if you know something about the number of electrons, because those are the subatomic particles that do the interacting. This leads us to discussion of what are covalent bonds, what are ionic bonds, right? And if you don't know the difference, you better ask or get studying because I'm going to expect you to know the difference between a covalent bond and an ionic bond. They're very different.
How many questions are going to be on the test or the quiz?
The chemistry?
Do you have an idea?
30 multiple choice.
The chemistry, 30 multiple choice. Yep. For the lecture and lab quizzes tomorrow, I think there's somewhere between like 8, I think there's like 8 questions on each quiz, something like that. So most of the questions are worth 1 point. There's a couple worth 2, I think. But they're short. But the chemistry, 30, each worth 1 point apiece. Yeah.
Yeah, how I've always remembered like covalent bonds is like co-parenting, like they share, you know? That's one way I remember.
Right. That's a very good way to think of it because sharing, we're talking about the two elements sharing their valence electrons, they're called. Those are the outermost electrons. So you have to understand how the electrons are configured right within the atom. There's different shells that hold different numbers of electrons. The first shell can hold a maximum of 2, and the second and third shells,
and then
8. Yeah.
8.
Right. And we're not going to go much above the third shell, but you could talk about the fourth or fifth. It depends upon what element it is. And the goal really is to try to fill your outermost shell, if you can do that, because that's a chemically stable scenario. And so one way to do that would be to share, as you say, with another element, and then you can both fill your outermost shells. Or in the case of an ionic bond, you can transfer or donate electrons from one element to another.
Electron.
That's called an ionic bond. And the result of that is to form charges on each part of the compound, and that's what holds that salt together. Typically, salts are produced thanks to ionic bonds. And we can talk about bonding if you have questions about that. Now's the time to talk about it. Again, you guys got to give me some feedback because I'm hearing silence, and that's not always a good thing. Because I'm assuming if I hear nothing, is that you're good with it.
I seem to be all right with it. I'm just looking at the other notes I wrote to see if there's anything that ever crossed my mind.
Yeah, that's what you should all be doing.
Sir, I was going to ask about, this is Ryan, with the characteristics of life, are you gonna expect us to be able to, like, on the quiz, because the way I've been studying, at least for this, is I've been using the study guide where we're actually filling it in, right? We're filling out the data, because I felt like that was probably the easiest way to, like, use that chart or that series of things, because that's got the most information on it. You're filling it all in. If you know how to fill it all in,
yeah,
you're going to be good for the quiz. So are you going to want us to be able to write out and then define all of those characteristics of life? I mean, I guess I'm just trying to figure out how to know, what kind of level of detail are you expecting us to be able to recall on this test?
Yeah, that's a good and a fair question, Ryan, to ask. It's a good one to ask. In the case of the characteristics of life, or even the requirements of organisms there that they discuss in tables 1.2 and 1.3, I am not going to ask you specifics on any of those tables. I think it's helpful for you to read that over and get a flavor for what they're talking about. But to be perfectly honest, I am not going to ask you what are the 10 characteristics of life. I'm not going to ask you that. I'm not going to ask you what are the 5 requirements of organisms. What I am going to expect you to talk to me about in some detail are those topics that we did spend some time lecturing on, or Zooming lecture, right? That's homeostasis, that's positive feedback, that's negative feedback, that's knowing the 10 organ systems or 11 organ systems, which I didn't lecture on, but I think I said know these. So there's quite a bit in the first part of chapter 1, where it talks about the origins of medical science, learning A&P. We did talk a little bit about levels of organization, so that would be something certainly to take a look at on page 12. But don't worry about memorizing the characteristics and requirements of organisms. Yeah, don't worry about those.
Perfect. Thanks for the clarity.
Yeah, and thanks for asking. And that's what you should do. Seek clarification so you're not wasting your time memorizing something you don't need to know. But homeostasis is such a critical concept. You're going to hear about it every chapter. So you got to know what homeostasis is. You got to know the role of receptors, control center, and effector. Critical, critical, critical. Got to know that.
What about the whole chart for the underlying mechanisms?
What page?
I made a flashcard. It's like pressure gradient, concentration gradient, diffusion permeable, that thing.
I don't know where that is.
I believe that's section 1.6 in the textbook, I believe. That's at least what I have from my notes. or concentration gradient. Wait. 1.3 and 1.4, I think,
Okay.
section. In chapter 1.
Okay. Oh, yeah. That's 1.4. I see what you're saying. Underlying mechanisms and processes, foundations of understanding. Again, I would not be memorizing gradients and permeability, cellular differentiation. No, don't worry about memorizing those things. I would like you to read that over and try to conceptualize what they're trying to get at. This is going to be the first time you have been exposed to some of this stuff, so it's not going to make a heck of a lot of sense. But we're going to hit those topics again and again and again and give you examples. So you can come back to this section in December when you're done with the course, and you can read that over and say, oh my gosh, I understand what that all, how it all fits together now, but I didn't when I first read it back in September 2nd, you know? So no, I would not worry about memorizing those mechanisms and processes, but just read that over. And what they do, of course, in the book is they take terms and they boldface them, right? The important kind of key terms. Those would be things to pay particular attention to, because you'll hear about those words again and again and again. I think it's important to kind of, again, read over those core themes, but I don't expect you to memorize those there on page 14 and 15. Chapter 1 is, again, an introduction, as the title implies, to give you kind of an overview, very, very general, broad overview, but it's a good one. Homeostasis, important. Levels of organization, important. Organ systems, important. And there are significant chunks of that chapter, remember, that we said we were going to do in lab, which we did last week, specifically body cavities, thoracic membranes, anatomical terminology, right? That was all lab stuff. But the textbook does put it in the last part of chapter 1. How about other questions?
So from what it looks like, from lab like 4 to 11, there's no print offs, and then I think 12 starts more print offs.
I'd have to go in and look in Blackboard. I'm not saying that every single lab is going to necessarily have a handout to bring each week. It depends upon the exercise. So if we go to content and we go to stuff to copy and bring to lab each week. And again, I can show you this. You all see the, I don't know, what are you seeing on your screen?
Blackboard.
Blackboard, okay. Why am I not seeing it here? Okay, so stuff to copy and bring each week to lab. And you scroll down through here. So this is, do you see the rat dissection diagrams that I'm pointing to?
Yeah.
Yep.
Okay, so that's obviously next, or this week. And then you have to check the syllabus to see what we're doing next week, which is 5, 6, and 7. So here's 5. And it appears, as you say, there's nothing for 6 and nothing for
Did you post 5 for us to see? Because I don't think I saw it on mine.
Oh, yeah, okay. It is hidden. We haven't done it yet, so these are all hidden. You can't see these in your course shell. This is my instructor course shell. That's why you see it here, because it's mine. I will be freeing this up. So I can go in here and I can say edit, and then I can say make available to all viewers and hit submit. And now if you go in, you're going to be able to see 5. Okay. And I thought, well, what I should probably do is just free all these up so you can print them all off like I had suggested earlier. So I'll do that.
Okay, thank you. That's why I just wanted to double check was all.
Yeah, I'm glad you said that. I guess I had thought I had freed up these because I know I told you guys to go ahead and just print them all off. So as I'm doing that, how about other questions?
Well, sir, just to clarify, you want, so you, for the lab for this week, then just to clarify, you're asking for the exercise 5 materials to print those off and the rat dissection diagram. Is that correct?
No, we're not doing 5 tomorrow.
Oh, okay.
The question that somebody asked previously was there's nothing on, what did you say, 5, 6, and 7? And because I had not freed these up for you to view, you didn't see anything. So there is 5 to bring with you a week from Thursday.
Okay, so then just tomorrow all we need is the rat dissection diagram.
Correct. Correct.
Okay.
Yes, in your lab book, right?
Right.
For
Obviously.
yeah.
Mm-hmm. Got that.
Yep. 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 How about other questions? Chapter 2, Chapter 3.
I have one quick question on the cell diagram. So obviously, I mean, there is a ton we could talk about with respect to the cell, and you could question us on. So I guess I'm trying to get an idea, like we're talking about, like, obviously you're going to want us to be able to label the structure and things like that off of the diagram and things like that. Is there, I mean, I guess what I'm trying to get at is what it's similar question to what I was asking about the life, the characteristics of life.
Yeah.
How much level of detail are we looking at here?
Yeah. Okay. So do you see the table in front of you?
Yes, sir.
Okay. So you're absolutely right. You know, when you look at a composite eukaryotic animal cell like you see in front of you, there's a heck of a lot of stuff packed into that cell. And no cell looks exactly like this, but it's just the artist's attempt to throw everything in there that could be in there theoretically, you know. Now, do most of our cells have a nucleus? Do most have a Golgi apparatus and a bunch of mitochondria and smooth and rough endoplasmic reticulum? Yes, they do. Do all cells have flagella? No. The only flagellated animal cell is a sperm cell. Do all cells have cilia? No. The fallopian tubes are ciliated and your trachea is ciliated and upper respiratory tract is ciliated. So this is just throwing everything in there that could possibly exist in one of your cells, keeping in mind that the vast majority do share a lot of these. The listing here on table 3.2, where, you know, goes through the organelles, kind of their shape and what they do. You noticed in chapter 3 PowerPoint, the PowerPoint Zoom, I did not spend time going through each of these separately for a number of reasons. Number one, you would have fallen asleep within 10 minutes. And number two, you have heard of a lot of these things, I hope, at some point in your past educational history, right, in high school. So I think you should know what the general function of the Golgi apparatus is. I think you should know basically what ribosomes do. Again, this would lend itself very nicely to flashcards if you want to, learn them that way. Yes, there's a lot of material here, just in that table alone. But it would be good to have some fundamental sense of what each of these subcellular structures are. I'm not going to ask you to label a diagram like this, although I think you ought to recognize a mitochondrion. I think you ought to recognize a nucleus. You should begin to learn what the difference is between the Golgi apparatus and the rough and smooth endoplasmic reticula are. The Golgi apparatus is sort of a standalone set of membranes, like a bunch of pita breads all stacked together. This is a cutaway view you can see here, right? And the smooth and rough ER, they basically extend from the nuclear envelope, as you can see here, and extend out into the cytoplasm. And that's why I didn't spend any time specifically going over those organelles. We picked up specifically here with the cell membrane, because I don't think you probably have learned a lot about the cell membrane in high school bio, probably. I mean, you know that it's the outer skin, if you will, of the cell, but how many of you had ever known about the phospholipid bilayer? I don't know, maybe some of you did, but I suspect a lot of you didn't. So that's why, again, I sort of didn't lecture on organelles. I thought you had had a lot of that before, and it was just a matter of going in and kind of reviewing some of those.
So to summarize what you said, basically you're asking us to basically have a general familiarity with the structure and function of the things within the cell, but focus more on the things that we wouldn't have learned necessarily within the high school chemistry. Basically, whatever you focused on in your lecture, that's really what you want us to focus our time on.
Yeah, in essence, that's exactly right. I tend to base the quiz questions and the exam questions on material that I spend time lecturing on. However, that is not to say you could not see a question on organelles, because I had asked you to go back and review that, but it's not going to be an over, how should I put it? There's not going to be a super great focus on a quiz on something that I just asked you to kind of look over. I'm going to spend more time testing you on stuff that I spent time, describing and explaining. But again, don't dismiss this sort of stuff. The organelles.
Right. Thanks. I think that answers my question. So
yeah, the vast majority of the quizzes and lecture exams are going to be over things that I spend time talking about. Or if I say to you, read over the section on apoptosis in chapter 3, like I mentioned earlier today. Just because I don't lecture on it doesn't mean you couldn't see a question if I asked you to look at it. I did notice when I went back and replayed that Zoom recording for chapter 3 that there was somewhere in this recording, I don't remember where, but I had tried to play the video and only the audio came out because I had to stop share. I would have had to have stopped shared and reshared the video, and I figured out kind of what happened there. And I tried to prevent that from happening, subsequently. But there is a point in the chapter 3 where you hear the audio and you're like, where's the video? Well, I didn't know that I needed to back out like I'm having to do here on occasion. That's why I'm always asking you, do you see this? Do you see this? Because I'm always wondering if you're not seeing it, and therefore I have to go back out and reshare. And I don't like that about Zoom. It's kind of a pain in the butt, to be quite honest. And maybe that's just the way the system works, but it it is a little bit difficult for me to have to flip back and forth constantly when I'm opening up, subsequent folders within folders. And it is what it is, I guess. So I will try to avoid that. But I did note that particular video that you heard the audio of, you can just go in here and click on the hyperlink. I forget which video it was, and watch it too. All right, it's not like you can't see it. You're just not going to see it during my Zoom recording. Does that make any sense?
Yes.
Okay. How many have looked at the chapter 3 Zoom recording? Somehow you guys can go in and raise your hand here, Where is it? Under,
I haven't looked at the Zoom recording yet. I just did the notes from the textbook.
yeah, under reactions, you can put the little thumbs up. Yeah. Good. Yeah, we might as well learn how to use those. We won't use this one a whole lot, I hope. That must be crying or laughing, I'm not sure which. Let's hope it's just laughing so hard you're crying. I'm sure they'll increase the reactions with subsequent versions. We'll be able to celebrate, after you get your quiz back, you get 10 out of 10. You can put that one up. That's great. I wish they had scratch and sniff, icons. So like when you get a 10 out of 10, you get like chocolate shake, scratch and sniff. And if you bomb the test, you get like vomit or something like that. I think that would be kind of fun.
Can we do that for ourselves, sir? Because that's probably what we're going to have to do.
Got a few minutes left. Anything you want to ask about chapter 1, 2, or 3? Until you get that first quiz out of the way, you just don't know how much detail to put into studying. I mean, that's really what it amounts to, if you're honest, if I'm honest with you. You just got to take a couple quizzes and you get a sense. I'm not putting enough time in, or I'm not studying with enough of the detail. The problem, quote unquote, with a course like this is there's so much. There's so much, it just blows your mind. And I certainly understand that. And I wish there was an easy way to break it down. You just got to be staying on top of it. Do not fall behind. Keep up with the readings. Ask questions if something's not making sense.
Sir, based on what you said, it almost sounds, I mean, I'm the old guy in this class probably. So my thought would be that, in a way that, because I'm actually reviewing a lot of things, because this is obviously a review from me from probably about 20 years ago when I did bio for the first time. So a lot of this is like me going back and going over stuff again. But what I was going to say is, it sounds, based on what you said, that what we should do is, if you watch the lecture, at least this is the way I would probably do it, watch the lecture and then supplement with your book. Because at least that would be the hint that I would give everybody else going on from here on out.
I think you hit the nail on the head, Ryan. We talked a bit about this yesterday in the review session, so this is probably worth repeating, because somebody was asking, like, how do I study? Do you have any study tips? And I said, what I used to do in all of my science and even other non-science classes, the technique that I developed, and you all have to develop your own study techniques, right? The way that I found worked really good. I was going to show you one of my notebooks that I even have my A&P and my zoology and botany notebooks that I took notes in. I can't pull them off the shelf right now. I can't seem to remember where I put them. But if I were to show you what I wrote many years ago, I would have this narrow leaf, notebook like you guys probably have. And what I would do is I would take the notes that the professor gave in blue pen in my notebook as he or she was presenting it. And then I would, and this takes a little bit of time to learn, I would leave gaps, like 4 or 5 lines, blank lines in my notebook between major topics, or maybe even half a page, so that when I went home that night and I read over the stuff that he or she talked about, as Ryan says, I'm going home and I'm reading over the text, because the PowerPoints that I give you are very chronological. They just flow chronologically through the chapter, so you can't get lost. You really can't. And I'd go home and I would read over those sections that was lectured on, and then I would use a red pen and I would supplement the blue notes with the red text notes, which reinforced the information. That way, when I studied for the quiz or the exam, I had my notebook and I had everything I needed. I had the blue notes from the professor. I had the red supplemental notes that I had to go in and write out myself. But you've heard how the best way to learn is to, you hear it, you speak it, you write it. It reinforces that, makes those synapses connect better. And then I had everything right there. Now, am I saying that I never went back and read the chapter again or 2 or 3 times? No, sometimes I would have to go read this a couple times. But when it came down to the test or the exam, I had most of what I needed in my notebook. And that worked great for me. I was not one to highlight the book with the orange highlighter or green highlighter, yellow, whatever. I just never did that. But you all know that some people do. They just highlight the book and that's what they study and that works for them. That just did not work for me. I had to write things down. I wasn't a big note taker, note card maker, I should say. I didn't do the note cards. I just never did. My daughter, who just graduated from Gannon in occupational therapy, I think I talked to you about her the other day. I told her she should open up her own store. She could sell her note cards, make $1 million. She just made tons of them, and then she reviewed them and wonderful. That's great. It takes time to make note cards. For example, if you do like the cell organelles, maybe on one side of the note card you have mitochondria, and you have a diagram of the organelle, and on the back of the card you have the function, or whatever. There's ways to do that, but that takes time. But learning takes time. It can't be rushed. So you got to put the time in. But I think you're absolutely right, Ryan. Just following the PowerPoints and focusing on that material is the way to go. Yep. And review, constantly review. Years ago, one of the classes I had, I think it was a microbiology class, they had this made for me. I should bring it. I always say to myself, bring it to class the first day and just hit them over the head with this. It summarizes it, Review, keep reviewing. And then many years ago, I had three women in microbiology. They made a t-shirt for me. They said, "There is life after bio with me." And I used to wear that to class. That was a long time ago. That's funny. They're now nurses, I think, at down in Bradford. What's the hospital there?
BRMC.
What is it?
BRMC.
PRNC?
BRMC. Yep.
Was it called something else before that? No, Bradford Regional Medical Center.
Medical Center, yep.
Okay. Yeah, I think sometimes students walk into micro or A&P, like this course, especially A&P, I guess, because by the time they get to micro, they kind of know how to study. When you first walk into A&P, it's a little daunting because there's so much material. How do you tackle this beast, you know? And it takes some time to figure out what works for you. And you'll struggle with that a little bit, but you'll eventually get to a point where it becomes clearer to you how much you need to study or how detailed your notes have to be and that kind of thing.
Yeah, my stepmom was telling me, she's like, you really need to study in his class. She goes, I cried to him asking him to pass me.
Who is this?
Amber Delgado.
Oh gosh, the name sounds familiar. When did she take A&P?
It'd probably be 2 or 3 years ago.
Oh, that wasn't that long ago.
No, she's like, she has really dark hair. It's really curly and she wears a hairband all the time.
Okay.
Yeah.
Oh, she probably has a bunch of horror stories to tell you. Don't believe any of them.
Yeah.
Do you remember Brianna Mondriel? I think she's Filipino. We're the same ethnicity. She's an RN at Encouty now, I believe. I believe she said she took your class.
What's her last
I'm not sure how long ago. Mondriel. I think she got married, so she's Filipino. I don't know if you remember her.
I don't. Maybe she had her maiden name at that time, right?
Yeah, I think so. She said you were a really good teacher.
Oh, you're just saying that.
No, she said that.
I think a lot of people, I mean, there are people that just will tell you don't take A&P with Ratterman. I mean, there just are, and you probably have heard that. But I hope that more people say just the opposite. And I'm not trying to brag, believe me, I'm not that kind of person at all. But I will tell you that I've had a lot of students who come back to me who've been practicing nurses for many, many years, or maybe just a couple years, and say, I am so happy that you made us, you know, work as hard as we did, because it made nursing so much easier. And so, you know, I am not out to make your life hell. I really am not. But I am out to give you the tools to succeed in not only this course, but in A&P2 and micro and your nursing. And, you know, some of you will go on to get your BSNs and your, you might even be, you know, nurse practitioners, right? So this is the beginning of a long journey for some of you. And
when would we take microbiology? When is that? Would that be next year?
Well, you could take it after you take A&P1. So theoretically, you could take it in the spring if you wanted to. I would say most students going into the nursing program would probably complete the A&P sequence, A&P1 and then 2, and then take micro right after that. That could be in the summer. It could be like for you guys, maybe next fall, a year from now. But theoretically, if you said, I want to get micro out of the way and I don't want to take it during the summer, I mean, you could theoretically take it in the spring. The prerequisite is A&P1 or Principles of Bio 1. But I would say
yeah, I was thinking about taking summer classes. Do you graduate any sooner if you get the classes done, like say in the summer? Or do you still graduate at the same?
Well, are you going into the nursing program?
Yeah.
So I don't think that would speed you up at all. I think it would take away, you know, if you took the lecture and the lab both, you know, that's 4 credit hours that you don't have to worry about taking the following year. It just kind of gives you some breathing room, but then you're committed to 6 weeks of micro, which isn't the worst thing in the world, but you know, you're eating, living, breathing micro lecture and lab 4 days a week, basically in the summer. That's the downside. It's not a bad thing. Now, last summer, we didn't offer the lab because of COVID.
Right.
Hopefully this summer will be different.
Hopefully.
Yeah. Okay, it's 2:30, so I think.
Yeah, I took like.
I've got lab in 10 minutes.
Oh, you can keep.
Micro lab, so I'm going to have to kind of close the class here. Please try to come with questions next week. I will be getting the next chapter, which is chapter 4 on cellular metabolism. I'll start putting at least the first part of that chapter, the lecture in Zoom. And I will just tell you, this next chapter is probably, I won't say one of the more confusing chapters, but we're not done with chemistry yet. We're going to be getting into a little more chemistry in chapter 4. These are going to be topics that you, I think, have all heard of, but we're going to be going into maybe a little more detail than you're used to. I'm not trying to scare you or anything, but try to get into chapter 4 before next Wednesday. Okay.
All right.
Okay, guys, I will see you tomorrow. Have a good night.
Have a good day.
Thank you.
Thanks.
Professor Redmond.
Yeah.
Can I just ask you one quick question?
Sure.
For the lab report, the third column on the graph, because the first one was for the ulna length, and then the third one.
Right.
Can you just explain real quick, like, what that stands for?
Are you talking about where we had the one column of ulna length and then height and then the last column? Is that what you mean? You're muted.
Yes, that's what I mean. Sorry.
Okay, so the last column was height divided by ulna length.
Yeah.
And that value is different for each person.
Right. What's it stand for?
Okay, well, that particular number is a ratio, right? Well,
Okay.
what was our hypothesis ratio? Do you remember? For the skeleton? 5.9, right? So in the results or in the discussion section, when you talk about accepting or rejecting the hypothesis, you could theoretically look at those values and comment on, you know, how diverse they are compared to, say, 5.9, and that would be another reason why you might end up rejecting the hypothesis.
Okay.
But you're also going to be looking at those lines on the graph, right?
Right.
It's going to help you decide whether to accept or reject. But that column, that ratio, that fourth column of ratio numbers is basically what we did with the skeleton. We divided the skeleton's height by its ulna length. We did all your heights by all your ulna lengths. And so that can be another way of comparing.
Okay.
Does that make sense?
Yes, it makes more, because I was thinking like it was like the height in feet.
Oh, no.
I was thinking about it completely wrong.
Yeah, good to ask. No, that was where you took your height in centimeters
Mm-hmm.
and divided it by your ulna length in centimeters. And we all did that. And that's where all those numbers came from.
Yeah, okay. Thank you. I just wanted to clarify.
Oh, yeah, I'm glad you asked. I appreciate it.
Result discussion section wrong.
Yeah, I'm glad you asked, Kelsey. That's good. All right.
Okay, thank you.
You bet. See ya.
See you tomorrow.
Yeah.